Hi everyone, and welcome to this week's community Q&A webinar. If you're tuning in, it's been a while since we've hosted one of these through the Healy Center, but we're back at it. So if you go to the next slide, as part of this webinar series, we often get suggestions and recommendations from members of the community and members of our patient advisory committee. And the topic for this week was actually a direct suggestion from members of our patient advisory committee, who pointed out aptly so that sometimes scientific publications, or oftentimes, they're not very easy to understand if you aren't one of the scientists who wrote the paper. And so we brought in our favorite biostatistician to have as a guest speaker on these webinars, or at least one of them, very much in the top two at the very least. So Dr. Lori Chibnik is here with us today to go through some tips and tricks for how to make sense of these types of scientific papers, specifically in the context of the first five publications for the results for the platform trial. So with that, Lori, I will hand things over to you.
Thank you. I can't be upset about being in the top two. Um, but I'm excited to be here. I always look forward to getting invited to these seminars and having an opportunity to speak to people who are living with the disease and caregivers of those people and people who care about the community. As a statistician, I tend to spend a lot of time looking at numbers and talking to numbers, and sometimes they talk back, but we won't go there. And so it's always nice to be able to actually talk about our science to people that are living with the results of our science. So uh, I'm going to talk today about how to read through a scientific paper. And I'm going to use our five papers in the Healy study as an example. So I don't know why that is going the wrong direction. So what my plan, basically a way I approached this was, what are the Healy platform papers trying to tell me? Trying to answer that question. So we'll walk through the five papers with a critical eye, or at least with what I think is my critical eye and my suggestions on what things you might want to be looking for. And then throughout this journey, I'm hoping to also share some knowledge a bit about how to read published papers, like what to look for, what to ignore, what are good things and what are bad things. Um, but I'm also going to skip to the end before I even get started, which is, I was asked to do this to give some insight into these papers. And I just want to remind people one of the goals of the Healy trial, of course, our foremost goal is to find drugs that work, find drugs that slow disease progression, that stop disease progression. We want to find effective drugs. But a secondary goal of the trial is also to weed out drugs that don't work, weed out drugs that and medications and compounds that shouldn't go to phase three trial, that shouldn't waste people's time in a phase three trial. So part of the goal of the Healy study is to quickly go through compounds and say, this isn't worth the time and it isn't worth people putting their effort into it when they could be in other trials. So we want to make sure our phase three trials are really the ones that have the most chance of being successful. And in that regard, the Healy study has been really, really successful. I only say that because I'm going to talk to you about five papers that didn't meet what's called their primary endpoint. As you know, I think many people know, two of the compounds, two of our companies are exploring and looking to build phase three trials, which is great. But ultimately, they didn't reach their endpoint. But I don't think that this is necessarily a sad story or not a completely sad story, because these are now three drugs or more that aren't going to be enrolling people in phase three trials when we know that there's a very little chance that they are effective. So that's my skip to the end. We're not going to necessarily tell you anything new. Um, I also am going to encourage everybody to ride along with me. So on this slide, you have QR codes to get to all five of our papers. I am going to show a lot of screenshots. I'm going to hit on points on all five of those papers. But if you want to read along and you want to look at the papers as we move along, the QR codes are available here. And yeah, let's get going. So the first thing I was going to talk about is just a typical manuscript. We're talking about peer-reviewed published papers very specifically. They start with an abstract. That's always a short summary, and it's always free to see. That is something that is not behind a paywall. The abstracts of papers are always available to anyone. Then they are usually broken up into about 4 sections. Each journal has its own style, but in general, the sections are an introduction, which is why they did it, methods, what they did, the results, what they found, and the discussion, how they, the authors, think you should interpret their results. Those are the 4 general sections. Sometimes they're named something differently. And many journals nowadays, and it's becoming more and more common, will also include with a published article a lay summary. So written in a way that non-scientific communities can understand what's going on. And these are always free. And then an infographic, which is an pictorial representation of the results. And many of them even do podcasts as well. And I know moving forward with the Healy study, we're prioritizing when these papers come out that we include lay summaries. Even if the journal doesn't support it, we will include a lay summary. Um, for a couple of the articles that came out recently, we even had podcasts, which was really great. So there's more and more evidence that we're trying to translate these studies into an audience of people that maybe aren't, as ingrained in the scientific language. So what I'm going to talk about today, I'm really going to talk a little bit about the abstract, a little bit about the methods, and then we'll focus on the results. Those are definitely the areas that I spend most of my time in. So what I have on the screen, pretty much every slide moving forward is going to have a letter in the corner. So whatever letter that is in the corner, that is the study, that is the article that I'm showing a screenshot of. So this is the abstract for regimen A, the Zaluka plan. And you don't have to squint, that wasn't the purpose of this. I just wanted to highlight a couple of things. One of the first things you might see if you look at an abstract are paragraphs that look like this. This is the main outcomes and measures, is the methods section, and then the results. With abstracts, we don't have a lot of room. So we have to really condense everything that we do. And those results sections usually are just filled with jargon. So a scientist or a statistician like me will read this and understand what it is, but it is not the easiest way to understand what's going on in the paper for somebody who isn't a statistician or isn't familiar with all this, well, scientific jargon. However, there is a section of the abstract that is written in a very clear way, and that is the conclusion. For this particular journal, they call it conclusion and relevance. And this is what the authors say the result of the study is. And it's written usually in a very clear way. In this randomized clinical trial of Zaluka plan, treatments did not alter disease progression. This will give you the takeaway from this particular paper. And I want to emphasize that we're talking about published papers. So to get a paper published in a journal, you submit it. It gets sent out to usually about 3, sometimes more, what's called peer reviewers. And these are experts who are not involved in the study. And usually there's at least one statistician, a couple of clinicians, maybe some other basic scientists who read the paper, make comments on it. Then we as the authors have to respond to those comments. The reviewers have to be okay with our response, but sometimes there's a lot of back and forth. And only then the editors will make a decision based on the reviewers' comments whether that paper will get published. So there is a lot of back and forth before this abstract makes it to your eyes. So these are not press releases. Press releases are usually within 3 days of when the study's done. They don't have all that back and forth. They're written either by the company or we write our own ones. So there is, we do critique them obviously, but it's not critiqued from the outside view. And these really are to ensure that these conclusions that you see here in the abstract really represent a realistic understanding of what happened in this trial. They're not overly optimistic and they're not optimistic, which I think would be pessimistic. But a group of experts read this. They believe that this matches exactly what it is. These are also not third-party websites. There are many third-party websites that might try to interpret this study and come to different conclusions. But the ones that you'll see in the abstract and in the paper have been reviewed by a lot of people. It doesn't mean they're always perfect, but it does give me more faith that it is a realistic, fairly unbiased review of what the study's results were. I'm showing you this one from E. The previous one, I believe, was from A, only because depending on the journal it's in, different journals have different styles. So E was in a journal called The Lancet Neurology specifically, whereas A through D were in JAMA journals, which is the Journal of American Medical Association, and they just have different there's just stylistic differences. This isn't called conclusions, it's called interpretations, but it is the same thing. Okay, so let's dive into the actual paper. Every clinical trial that is reported in the literature will have this, which is called a consort diagram. Just shows you the flow of the participants. Um, it, I'm breaking it up into three sections. They're very standard. There's a single way to do it, although the formatting is usually different depending on the journal. But the top part will be screening, how many people were screened. For the HEALEY trial, we actually have an extra screening part because of the master protocol. Then it's the section of after they're randomized and how they flow through the study. And then finally, the last bit is how many people made it into the analysis. So this is just a way to see the flow of participants throughout the study, and it is very standardized. We also have baseline characteristics. So this is, most of us also call this table one. In most papers, table one is the baseline characteristics. And this is where you'll see a description of the people who participated in this study. So the columns will be active versus placebo groups. I'm using C as an example because it's one of ours where we had 2 doses. So you'll see that one column is for the 30 milligram dose, the other for the 60 milligram. And then in truth, the HEALEY is a little more complicated because, as you know, we have our regimen placebo, which is here in this column, and then we have the full placebo, so shared across the regimens. If I cut this off because of space, but the footnote will tell you that these 41 regimen placebos are also included in the column for the shared placebos because this was our comparison group. Usually when I look at table ones, I kind of eyeball. I want to make sure that we don't see a lot of differences. I don't expect to see any, but I like to kind of eyeball it. But maybe when you look at a table one of the questions I can see you asking is this what I see the baseline characteristics? We've got the distribution of male and female. We have the distribution of different characteristics of the disease. Does this represent what you think of as the ALS population that represents you? There might be studies that look at very specific populations. HEALEY doesn't. We had pretty wide inclusion criteria and exclusion criteria, especially for the first 7. But I might look at this and say, does this represent me? Is this representative of the ALS community? And it just gives you an idea of who's in the study. Okay, so let's get to the pictures and let's get to the nitty-gritty, which is the results, which is what everybody wants to see. So I'm going to walk you through this slide. I chose C as an example for one reason and one reason only, is that of all of our papers, this is the one that had the most contrasting colors. So I thought this would be the easiest one to look at. I'm joking about this, but it is worth noting that a lot of this formatting is determined, actually, all of this formatting is determined by the journal itself. So we did not get to pick the colors, but more importantly, there is a lot of back and forth, and it actually takes a lot of time to format the tables and figures in the way that the journal tells us to. So sometimes that is why there's a delay in getting these papers out, which is unfortunate. But let me talk you through our 2 main figures, because in our study, these are pretty common figures that you'll see in our paper. So let's walk through this with the axes first. So on the y-axis here, it says ALS FRSR score change. So this is telling me that this is the ALS FRSR score change from baseline. Again, I cut off the footnote. I apologize for space, but in the footnote, it indicates that it's changed from baseline. You can also kind of figure that out by looking at week 0, which is baseline, and everybody is 0 because that is change from baseline. On the x-axis, you have the week, and then each one of these was one of the visits, if you guys remember. And what I'm looking at and what I think is important to see are two things. One is the orange versus the bluish gray. So the orange are all the actives, and the gray or the blue is all the placebos. I'm less interested in the regimen-specific placebos. They aren't powered to see anything, which is a statistical term, so they're less interesting to me. I'm more interested in seeing the full set of placebos. And these little boxes indicate the average. So the triangle or whatever triangle or square in the box is the mean for that group at that week. And then the box in the middle is just the middle of the distribution. And then those whiskers, which is what they are called, are whiskers, just show you kind of up from the smallest to the largest. And what I'm looking for is, are these orange boxes changing at a different rate than the blue boxes? You'll see there's a line that is kind of mapped over it. This is a line that is produced by the statistical model that we use. It's a little deceiving here, but there's actually two lines. There's an orange line and a blue line indicating the active versus the placebo. But because there was no difference, those lines are lying right on top of each other. So it's really hard to determine, what color it is. So this is what we call our functional graph. So it's showing the functional progression. On the other side, we have something called a Kaplan-Meier mortality. And this is a mortality graph. So as you know, one of our outcomes besides function was survival. So a death or going on permanent assisted ventilation was considered, we use the term event. And the way this is situated is on the y-axis. It says mortality probability. It can be interpreted roughly as the percent who had the event at that time point. So that would again be a death or a permanent assisted ventilation. And then again, we have our orange and our blue line. And each time it goes up, each one of these little 90-degree steps is there was an event or it marks when somebody drops out of the study before they have an event, which is marked a little bit differently, but that's what these steps indicate. The lines, again, are produced from the model that we use, the statistical model, to model it. And again, even though these steps look a little bit separated, we are looking for those steps to kind of go like this. We really want it to go like that. These are kind of more in parallel. But when we plotted it with our statistical model, as you'll see, again, the lines are on top of each other. And that is represented in our effect measure, it's on the bottom of the slide, but I'm going to explain it more in the next slide. So I will jokingly apologize ahead of time that us statisticians used one of the more complicated models and the very not intuitive measures to measure the efficacy of our trial. I ask your forgiveness because it is the most appropriate model given the nature of the trial. So I'll walk you through it. The final effect measure from our model is called a disease rate ratio. And it's not something we see a lot. It's not something it's used a lot in clinical trials, but it's not something that's always used. And if you want to understand what it is, you can go to the methods section. So on the right is one of the screenshots of one of the methods sections. It will explain to you, and if you see the last sentence here, how to interpret this DRR. 1 means there's no difference. Less than 1 means the drug is better than the placebo. Greater than 1 means the placebo is better than the drug. So you can find how to interpret it usually in the methods section. The posterior probability of superiority is the probability that DRR is less than 1. This is different from our old friend, the p-value. For a p-value, lower is better. We're always looking for less than 0.05. This probability higher is better because you want the probability that the DRR is less than 1 to be very high. So I will show you from the others. So this is regimen A. As you can see, they're very similar graphs, although the editors chose different colors. And on the bottom, you'll see the DRRs and the posterior probabilities. This is A. This is B. And I'm going to pause on this for 1 second because even when I posted this up, I had to take a moment to look at it. I get that it looks like there's a difference because there's this very dark line right here. That's actually a weird formatting thing that the journal did. It's the line at 0. So 0 would be no change, and I think they highlighted it. The two lines for the placebo and the verdipastat are actually both on top of each other and going on the angle that matches those boxes. So I wanted to point that out. I'm also not 100% sure why that journal decided that this graph should be blue and gray and this matching graph should be orange and gray, but it is from the same paper and the same drug. This is D. As you see again, the lines are pretty much on top of each other. And then finally, E. So I'll pause for a moment on E because this was the one that was in a different family of journals, which is the Lancet, and they asked for a different formatting. They didn't want these. By the way, we call these box and whiskers. Statisticians are not that creative. It looks like a box and it looks like whiskers, so we call it a box and whisker. But Lancet wanted just these lines and then the dots and the squares and the points actually indicate the observed mean at that time period rather than the boxes and the whiskers. And you do see a little bit of separation where the gray is the placebo and the green is the drug. But now let's look at the other plot. Now, this is called a survival plot. It is the same thing as our mortality plot, just flipped. So instead of our y-axis measuring percentage of people who've had event, this one measures the percentage of people who haven't had an event yet. So every time it goes down, it's marking an event. So at this point, this percent of people are still event-free. You'll see in this case, the green line is actually going a little bit lower than the black line. So the actives are a little bit lower than the placebo, which indicates that there were more deaths, possibly faster, although not statistically different in the actives versus the placebo. And then those lines are from the model again. So those are our main primary results. And as I showed you, they were not significant, our primary analysis. However, we also have secondary analyses. So I'll talk you a little bit through these as well. And all of our papers do present these secondary analyses. These are different models. So we are back to our old friend, the p-value here. But I want to walk you through this table. They're formatted differently in every single paper. This was not our choice. This was based on the editor of those journals. So sometimes when you look at these, if you want to try to interpret these tables, you just have to pay a little more attention. So on the left here, we have our outcomes, and we're just going to look at the top half for a moment. ALS FRSR, strength, respiratory, etc. On the top of these columns, it says 24-week change estimate, and then SE stands for standard error. So what these numbers represent is, on average, in this group, how many points of decline did that group have for ALS FRSR? And it's controlling for other factors as well. So I would interpret this as, on average, those on active treatment lost 6.48 points over the 24 weeks. Those on placebo lost 5.61 points over the 24 weeks. These next columns are just the difference in these two numbers. And then the p-value is telling me the probability by chance. So basically, is it significant? And as you can see with these p-values, none of them are significant. The top half, and again, for space, I did cut off the footnote, but the footnote explains that the top half here is what's called FAS, so that's the shared placebo, and the bottom half is just the regimen-specific placebo. And so these tables, I'm not going to show you in each paper just for the interest of time, but these tables are in each paper. But I will show you, because we have one other secondary result to show, is for paper B. I'm embarrassed to say that I did create this table. I think it is very messy, but this is what the journal wanted. It is too busy, but I will direct your eyes to the bottom part of this table, which is the other outcome that we spoke about, which is what we call survival. So this is time to an event, which in this case, again, is death or permanent assisted ventilation. And these are called survival models. The survival model, their effect measure is something called a hazard ratio, but you can interpret that as a risk ratio, and I'll interpret a couple of these for you. So again, in our full analysis set, the model that controlled for other factors, there was a 1.54 increased hazard of death or PAV for those on the drug versus those not on the drug, but please, take note of the p-value being very non-significant. I said hazard, but you can think about it as 1.54 increased risk. There is a difference between a hazard and risk, but it's very subtle and mathy, so you don't have to worry about it. So anything above 1 means an increased risk for those on the active drug versus placebo. We are hoping for a below 1. But all of these are presented in our papers as well. So what else is there? And I only had about 20 minutes to speak, so I tried to cover the topics that I thought would be of most interest. So we also sometimes have post hoc analysis. Post hoc means we didn't plan for them, but once we got the data, we wanted to look at them. We tend to not put those in our primary papers, but sometimes there are additional analyses, exploratory, secondary, that we'll also put, and those might be different across the regimens. And then there's supplemental materials. The age of the internet allows us to have a lot of supplementals that you can download from the website. Additional information on the methods, additional results that didn't fit in the paper because there's only so much you're allowed to put in, maybe some post hocs. Sometimes the reviewers really want, like, have this question and you respond to it, and that whole situation is put into the supplemental. And then we also include our official statistical analysis plan and protocol. I also want to comment, and I meant to comment on this earlier, is that, as we said, these are peer-reviewed papers. They do take extra time because of the peer review process. We strive very hard here at Healy in the trial that all of these papers should be accessible for free and not behind a paywall. However, sometimes the journals, it is their decision because once we submit it, the journal then owns the paper. And we don't always win these fights. And sometimes it is the journal itself that puts these papers behind paywalls. That being said, if you want access to this paper and it's behind a paywall, I strongly encourage you to reach out to your clinician or really the clinic. They can get you that paper and send you a PDF of the paper. It takes us 10, 15 seconds to be able to do it. Okay, maybe 30 seconds if we don't type that fast or whatever. But we're always very, happy to send people that paper. We don't want it to be behind a paywall, but sometimes that choice is not ours to make. And then finally, if you get overwhelmed, and I know I just presented a lot of stuff, and I know there was a lot of numbers, and I know there was like figures and all the stuff that us annoying statisticians like to do. And I don't want you to feel overwhelmed because the bottom line is part of writing this paper is being able to put it in words that people can understand and not just our colleagues. So I encourage you to look at that conclusion section. So I just have 2 examples here. I chose these 2 because in the JAMA papers, the conclusions have the big title to it. In the Lancet paper, it doesn't. But if you look at that last paragraph, it usually starts with the phrase "in conclusion." And these should be written in a language that everybody can understand. So I encourage you, if you feel overwhelmed, to just look at the conclusions and then ask questions. We're always happy to answer questions and try to clear up. Catherine is easily one of the most approachable human beings I've ever met, and she's always happy, and she always passes questions on to me, and I'm always happy to answer them. With that, I'm going to stop talking. I'm very happy to answer questions. I needed to find that. but I needed to find that, so I now have it. But I'm happy to answer questions. I see one of them. But Catherine, would you like to promote the future webinars while I take a sip of my tea?
Sure. And thank you, Lori, for that amazing overview. I think I always feel like I just got like a college level educational course for free. And not coincidentally, you do actually instruct people regarding statistics. So thank you for being here and for putting together the talk today. For November, we are back on schedule with our 2 webinars per month. So as usual, the second Thursday of the month, November 13th, we'll do an expanded access EAP discussion. And then we're getting in just under the wire before Thanksgiving. So November 20th. I think we bumped it up to the third Thursday, so it's a little bit earlier, taking into account the holiday, but we will be having a platform trial webinar discussion on November 20th. So I'll put the link in the chat box here for people to register for those.
I do see a couple of questions, so I will answer them. Can you further explain the concept of shared placebos and how that concept has been important to the Healy trials? That's an excellent question because we do use a lot of terminology and I want to make sure that we're clear. So as many people are familiar with the design, uh, a participant gets randomly assigned to one of the enrolling regimens, and then those regimens are 3 people on active for every 1 person on placebo. It's very important to us to keep the number of people on placebo as small as we can while still getting useful and making sure that it's scientifically valid. But that does mean we have a lot fewer placebos. And if we were to just analyze this 3-to-1 randomization, it would be 120 on active and 40 on placebo. We would not have enough what's called statistical power to really see a difference. There would not be enough signal over the noise, if you've heard the expression signal for noise. So the platform trial really is a great device to be able to increase those number of placebos while not increasing the placebos in each regimen. So we are able to share the placebos across multiple regimens. So A, C, and D, their shared placebos include all the placebos across A, B, C, and D. B chose to just include A, B, and C, so I feel the need to differentiate. But they were able to basically say, okay, in A, I have 120 people on the drug and 40 people on placebo. However, when I analyze it, I can analyze 120 on drug and 160 on placebo because I have the placebos from 4 different regimens. So it increases our statistical power without having to really put more people on placebo than would have to be on placebo. Um, I hope that, and so when we say shared placebo, that's what we mean. It's that big group of placebos together. For F and G, they only shared from each other. There's a lot that goes into how and which regimens share with each, but we always make sure we have multiple regimens so that we can increase that placebo number. And that actually feeds into the second question that I see, which is the aggregated results inform us. I think there might be a word missing, so I'm going to interpret this as, do the aggregated results inform us about the usefulness of the shared placebo? It's an interesting question. I hope I'm interpreting it correctly. Um, I do think it does. So we have one of the things, and the FDA is concerned about it, and we're concerned about it. So it's a fair concern. Is, can we share these placebos? Like, is this scientifically accurate? Is it justifiable to share these placebos? As the different regimens had different drugs. One had an infusion, one had an injection, one had a pill, one had a powder, right? Can we really combine them? So again, one of the beautiful things about this study is that because every regimen follows the exact same protocol, visit on week 3 or 4 and 8 and 16, and you get these measures at the same time, it's done at the same sites with the same investigators. It really has made our placebo groups very, very homogeneous, meaning similar. And we have compared the placebos across each other. So the different placebos we do what's called sensitivity analyses. So when we do an analysis with just the regimen, so the 120 versus 40 for the first 5, and we had larger numbers for the last 2, that's a sensitivity analysis. We're really testing to see if we should share the placebos. We compare the placebos across to each other to justify that we can share them. So we are able to do that, and we do take that very seriously, and the FDA takes it seriously. And we have looked at them. We've looked for time trends. Do we see differences in the people that enrolled 3 years ago in 2020, where the world was very different than people who enrolled in 2023? And we haven't. We've really, really dove down that. That is not included in our papers, but we have. I hope that answered the question. I see another question is it possible to be part of the HEALEY trial from Europe? We do not have sites in Europe, and that's the best I can answer. I would punt that usually to one of our clinicians who unfortunately isn't here today, but my guess is we do not have sites in Europe at this point. There are other clinical trials that are happening in Europe, and I would encourage you to reach out to Catherine, and I'm sure she can connect you to people that would have a better an answer to that one as well.
Yeah, and
Um,
I'm happy to chime in too, Lori, to just add
mhm,
that for the Healy platform trial, in terms of the inclusion and exclusion criteria, so Lori, you're right. All of our research centers that we work with are based in the United States, and because there are required in-person visits, and those in-person visits are fairly regular, like Dr. Chipnik was saying, week 6, 16. So it's over a good period of time that you have to be going back and forth to a site. So there's just added considerations there. It's not an exclusion if you live outside of the US, but it does add a lot of complexity to someone who wants to participate, and they do have to be able to travel regularly to in-person visits. So, um, I am happy to help navigate that conversation, but we do often advise, if there's a trial closer to home, um, that would be more feasible. But we can definitely have those conversations offline too.
right. That is the list of questions I had. I'm happy to wait a moment if other people have other questions. I want to thank Catherine and the Healy team for inviting me again to speak with you all. It's always a great honor to be able to actually communicate our science to the people who are living with it.
And I will thank our patient advisors and patient advisory committee for encouraging me to reach out to Lori Chibnik yet again to have you back at these webinars. It's not the first time we've had you here. It won't be the last. And there is, a compliment in the chat already saying amazing job to you, Dr. Chibnik, and thank you for hosting this webinar. Um, and with that, I think we've gotten through all the questions. So I will just say thank you again, Dr. Chibnik, and see you in November to those who are joining us in a couple weeks.
Thank you.